Greetings to everyone. I am Chrislene Eroy from Tagum College, Tagum City, Philippines, together with my colleagues Aiza Camporedondo, Jennifer Velos, and Ms. Karyn Chrislene Vitor, presenting to you our research study entitled Beetle Diversity in an Agroecosystem of New Bataan, Davao de Oro. We are very excited about this study and we hope to deliver the salient information about this biodiversity research. So to start, what do we know about the beetle species? The Coleoptera order is the world's most varied insect order with an estimated 400,000 species. Beetles are beneficial to ecosystem durability by providing food and habitat to native species and providing vital ecosystem services such as pollination for reproduction, decomposition process, control populations of pests and insects. Furthermore, the International Union for Conservation of Nature reported that over 18% of saprosylic beetles in Europe are threatened with extinction because of the depletion of trees as their habitat. Logging, wood harvesting, and other sorts of tree loss are identified as the largest risk to threatened species. Pursuant to the Republic Act 9147, in support and to promote scientific research on the protection of biodiversity, this study was pursued. With this, our main research objective is to identify the beetles up to species level and to determine the species abundance, richness, and biodiversity. The site was selected in a coconut-based agroecosystem in Davao de Oro in a private area in Purok 13, Barangay Kamanlangan, New Bataan, Davao de Oro. Entry protocol was obtained from the tribal chieftain in the local of the study and wildlife gratuitous permit from the DNR Region 11. Communication with the nearby community was made for public information such as placing signage or the tarpaulin. Next is the establishment of the study site. A transect line was established along a 300-meter area and sampling quadrats were plotted. Each quadrat is 10-meter wide and 10-meter long, established alternately along the transect line with 100 meters interval. For the collection of specimens, sampling was made from 6 to 11 in the morning and 1 p.m. to 5 p.m. in the afternoon. Also, the following techniques were used: pitfall trap, beat sheet, and hand-picking techniques. So for pitfall traps, beetles were captured in plastic containers that were buried at 9 centimeters soil depth with the cover cut to 10% of its original size to allow passage and prevent trapped insects from escaping. Each trap contained 20 grams of animal feces in 48 hours as bait to attract beetles. The bait were wrapped in a fine cloth and secured inside the trap for simple disposal and to prevent insects from tampering with them. In beach sheeting, this technique was accomplished by shaking and pounding the tree or a branch while holding the beating sheet. Specimens fell into the sheet and extracted by hand or with forceps. In hand-picking, individual species were hand-picked as observed along study lines and plots. For the classification and identification of specimen, the specimens were photo-documented, including the dorsal, ventral, and dorsolateral view of the species at 300 dpi standards. Vulture specimens were thoroughly cleaned and preserved in a 70% ethyl alcohol and were transported to the laboratory of Tagum College for further identification. Morphology of specimens were used to initially identify the species using field guides and various manuals for entomology and Coleoptera species. Verification of identified species was made by an expert thereafter. For the experts, we have communicated with Coleoptera experts for the verification of the beetle specimens. Photo documentations were sent via email. We have two international experts, Dr. Zibajne Kejval from Museum Chodska, Domalslav, Czech Republic, and Dr. Dennis Keith from Museum des Sciences Naturales et de Préhistoire, Chartres, France, and one national expert, Dr. Alma Mohagan from Sibrim, Central Mindanao University, Bukidnon, Philippines. Next is the analysis of the recorded data from the sampling. Shannon-Weiner Diversity Index and Velos Evenex Index were used for the diversity indices. Margalef Index, on the other hand, was used in calculating the species richness, and PASS software was used to execute standard biodiversity analysis. There are a total of 294 individuals representing 6 families, namely Curculionidae, Chrysomelidae, Scarabaeidae, Antecidae, Phasalidae, and Hysteridae. Presented in this graph, the accounted individuals of beetles per family in the study area. There are 102 individuals from Chrysomelidae, accounting to 44.5%, 97 individuals from Curculionidae, accounting to 42.4%, 10 individuals from Phasalidae, accounting to 4.4%, 8 individuals from Hysteridae, accounting to 3.5%, 8 individuals from Antecidae, accounting to 3.5%, and 4 individuals from Scarabaeidae, accounting to 1.7%. We will be discussing each family in the next part of our presentation. Members of the family Curculionidae are easily recognized because of their long snout and geniculate antennae with little clubs, have a wide range of form and size, with adult lengths ranging from 1 to 40 millimeter. There are 3 identified genera belonging to this family. One of these is Rhynchoporus vulneratus. It is large in size, its head and abdomen is generally black in color, while its pronotum has longitudinal markings in the color red-orange. Rhynchoporus ferruginus, on the other hand, is smaller in size than Rhynchoporus vulneratus. Rhynchoporus ferruginus is characterized by black spot markings on the pronotal reddish body color. Metapocyrtus apuensis femor's primary color is reddish-orange, while the mid-femur and tarsus are densely pigmented segments of black. Its body is generally black in color with green to yellow scale stripes. Odoiporus longicolus, on the other hand, are dark to reddish in color and range in size from 23 to 39 millimeter. Species in the family of Chrysomelidae generally have an oval-shaped body, flattened dorsally and convex laterally. They also have a small head, antennae base that are separated by a keel in the front, and compound eyes that are slightly protruding. There are 2 identified genera belonging to this family, Podontia quatordecipunctata and Chrysolina species. Podontia quatordecipunctata has an oblong-sized body. The head, prothorax, antennae, and legs are shiny. The mouth parts are black, and its elytra has a bright salmon pink color, and each has 8 spots. Chrysolina species, on the other hand, has a long filiform antenna with 11 segments. Its pronotum is wider than long, and its abdomen is semi-oval, light brown with 4 sternites that are visible ventrally. The majority of scarab are uniformly black or brown in hue. Its head not deflexed or just slightly deflexed, and their antennae has 10 segments with 3 to 7 segmented opposable club. Cusquenhera sulcipenis can be described as having a mandible with a patch of tiny granules, a maxilla with 18 acute stridulatory teeth lined up, and each femur and tibitarsal with a cluster of fossorial setae ventrally. Members of the Antecidae family can be easily recognized by their heads, which constrict immediately in front of the pronotum, forming a neck, and the posterior end of the pronotum is usually thin as well. The ant-like appearance is enhanced by the short legs and antennae, and the body is sparsely covered in long setae. Antilephala can be identified based on their head, which is 1.2 times longer than it is wide. Its thorax and pronotums are 1.5 times as long as wide and noticeably narrower than the head, including the eyes. Phasalidae, on the other hand, is the family of shiny black beetles with their size ranging from about 1 to 8 centimeters in length. Members of the family Phasalidae feed on rotting logs, typically seen in moderate numbers of larvae and adults. Leptolux species is relatively large with a black glossy color. The pronotum, antennae, and middle sets of legs are lined up with golden hairs visible to the naked eye. It has lamellate-shaped antennae, small but thick mandibles, and a single curved horn points forward on top of the head and between the eyes. This Turidae family has distinctive geniculate antennae with terminal claws and truncate elytra, which will reveal the last two abdominal segments. Pachyluster species has a body length of 3.2 millimeter and 2.3 millimeter in width, and elongate oval-shaped body with black and dark brown color. They have shorter elytra, which exposes two of the seven tergites, and their geniculate antennae with club tips that make them easily recognizable. In this study, there are 9 identified genera and 1 unidentified taxon for the total of 229 individuals. The unidentified specimen needs to be verified further from the national expert. The most abundant species encountered is Metapocertus species, representing 32.75% of all individuals identified, followed by the Chrysomelidae species with 31.88%, while the least abundant species is Chrysomelidae species with 1.75%. To provide information on the population structure, richness, and diversity, we would like to present the Alpha Biodiversity Index at the Agroecosystem of New Bataan in 2022. We have the Simpson Diversity Index with diversity value of 0.7722, Shannon-Weiner Diversity Index with diversity value of 1.799, Velos Evanes with diversity value of 0.6046, and Margalef Index with a diversity value of 1.656. The agroecosystem of New Bataan, Davao de Oro is composed of Theobroma cacao, Musa acuminata, Cocos nocifera, fern allies, and unidentified grasses which may provide shade, nutrition, habitat, and interaction with other species. Beetles are important species in any ecosystem. As a pest, it affects multi-trophic level interaction, including the interplay between below-ground and above-ground herbivores via changing responses of plants to herbivores with important consequences for plants and higher trophic level biota. Pesticides are commonly used to improve agricultural output and prevent the spread of infectious diseases. However, negative consequences were reported such as increase in resistant pest population, a decrease in beneficial predators, and pollinators. It has shown pronounced negative effects on the biodiversity, not only in the agroecosystem, but as well in the small streams affecting the invertebrates, soil invertebrates, and soil microbes, along with contributing to water quality. It is essential that the complex ecology of the beetle species and other insects are well understood so that agricultural system as a whole will be improved. It is also a recommendation that farming techniques should be sustainable to support and sustain a biodiversity-friendly farming system, such as organic farming, reduction and withdrawal from harmful pesticides, and improve shade tree management. These are our references. With that, we would like to thank the Davao del Norte State College for the opportunity to present our study in this International Research Festival 2022. That would be all. Thank you and God bless.